welcome all of you either back or welcome here to this second series of delegate lectures by Bialek. The title of this talk is Signals, Noise, and the Physical Limits to Biological Function.
Okay, so for a long time, I've been interested in situations where biological systems perform extremely well. And there's a tendency to think of on the one hand, the phenomena of life are very beautiful. On the other hand, relative to some of the things that we're used to in physics, there's a tendency to think of them as a bit messy and certainly complicated. And what I'd like to do today is to take you on a tour of several phenomena where I think that impression of messiness can be combated. And we can uncover, or our experimentalist friends have uncovered, a surprising degree of precision. And in some cases, I think we've even succeeded in taking the observations of precision and turning them into something that at least is the beginning of a theory from which we can derive things in the way that we're used to in physics. So I'd like to take you on a tour of some examples. So let me start.
Could you give examples of some of the bad examples first?
So, yeah, it is very often so the kind of thing that drives us crazy is that if you look at biological systems, you often see complexities that seem unwarranted. So, for example, and maybe I'll loop back and solve this one for you. So for instance, many protein molecules, in order to they do various things, and their function is changed by attaching a phosphate group to them. That's not so bad. And it's actually not totally implausible. A protein's a polymer of amino acids. If you look through the table of amino acids, you don't see anything that's very highly charged. I mean, there's a little bit, but nothing like a phosphate group, which has four negative charges in a very small space. So you can imagine that if you take the structure of a protein and you stick 4 charges in a very small space, you'll push things around. You're applying a lot of electrostatic forces. And the protein structure changes, and that has an impact. So that sounds like a perfectly sensible mechanism for regulating things. The problem is that there are proteins that have half a dozen places where they get phosphorylated. And sometimes in order to get from sort of point A to point B, it seems like you have to stick all of those phosphate groups on. That seems awfully complicated. What's that for? So hold on to that, and maybe I'll come back to it. I mean, this is, and this is the tip of the iceberg, right? Sort of everywhere you look, you see this kind of stuff. So how do you bring some order to this? Could you imagine a theory that predicted that there should be 6 sites, right? I don't know, how would you get that right? If you measure the concentration of certain molecules that are crucial for the function of a cell, and you measure the concentration of the same molecule in many cells that seem to be doing the same thing, you might find that those concentrations vary all over the place. And so you say, well, okay, it really is messy in that these things are highly variable. There's another idea to hold on to. So those are the kinds of things that worry you. So let's actually take the problem of variation in concentrations. So a large number of signaling processes in biological systems come down to changing the concentration of certain molecules. So let me give you 2 things to have in mind as examples. One from the world of bacteria, and one from the world of just the typical cell. So this is going to be about bacterial behavior. And the other is from the world of what might be called gene regulation. I'll say, since this is a physics math audience, I'll say what I mean by that in just a moment. So let me just tell you a couple of facts, and then we'll try to zoom in on what I think is the essential physics problem. So a bacterium, of course, there are lots of different kinds of bacteria. There are more different kinds of bacteria than there are anything else. At least in the living world. But let's focus on bacteria that are not unlike the bacteria that live in your gut, E. coli. If they're living out in the world where conditions are not as good as they are inside your gut, they need to swim around in order to find something to eat. And so they are little objects that are about micron in size, and they have, so this is maybe 2 microns in length, and then they have these flagella that stick out from the end, and they swim off in this direction, trailing their flagella behind them, to simply state as facts a series of really beautiful experimental discoveries. The flagella are more or less inert proteins, made out of more or less inert proteins that form a corkscrew shape, a helical shape. And they are driven at their base by a rotary engine. And so the bacterium has sticking out from it essentially a bunch of rotating corkscrews. And it's the world's smallest rotary engine. Right? It's a few nanometers across. And it's powered by the fact that the chemical potential of hydrogen ions, so the pH, is different inside and outside the cell. And these motors allow protons to fall down their gradient in chemical potential, and that spins them. And if you think that that's a peculiarity of bacteria, you should know that all of the ATP in your body is being synthesized by molecules that do the same thing. They let protons flow through them and they rotate, and during the process they manufacture the molecule ATP, which is the energy currency of the cell, and in particular the thing that you use to move your muscles. So it was an astonishing discovery that these things have rotary engines. If you look at them under the microscope, if you think about it, if you see a corkscrew rotating and you look at it from the side, it looks like it's waving. And so people assumed that that's what was going on, and that turned out not to be true. If life is good, they don't even bother making the flagella. They just go about their business. But if, as in your gut at the moment. On the other hand, if life is a little more demanding, then they need to find their way to food. And the way they do that is they have receptors on their surface to which various molecules can bind. And then they have an elaborate apparatus underneath that turns this into an internal signal. And these signaling molecules can then bind to the motor. And when they bind to the motor, they bias the probability that the motor turns clockwise or counterclockwise. Why is this important? It's because if you have corkscrews, and depending on the handedness of the corkscrews, you put 2 corkscrews next to each other, and you rotate them, and they have the same handedness, and you rotate them one way, then you'll notice that they fit together, and they stay interlocked with each other as they rotate. On the other hand, if you rotate them the other way, they keep banging against each other. And so correspondingly, when the motors switch from clockwise to counterclockwise, the multiple flagella of the cell either come together in a bundle and all rotate together and push the bacterium this way, or they fly apart and are all rotating independently of each other in which case the bacterium just sort of tumbles in place. And so what happens is when the motors are going in one direction, the bacterium swims, and then at some point it flips a switch, and now it tumbles around, and then the switch flips back, and it darts off in a new direction, whichever way it was pointing. So it's taking a kind of random walk, right? Because every time it tumbles, it picks a new direction. And it then biases its random walk such that if along its path, the concentration of these molecules outside that it likes is going up, it should keep going in that direction. So this is a version of a Monte Carlo algorithm that some of you may actually have implemented, right? If you're taking steps and the quantity that you're trying to, well, usually Monte Carlo is trying to decrease something, namely the energy. If you're walking along and the energy is going down, you keep going in that direction. But if you discover that the energy goes up, you should try going in a different direction. Okay? And this works. And in fact, bacteria are extraordinarily sensitive. They can respond to concentrations where the typical concentration in the environment of the molecules they're looking to eat are in the range of nanomolar, which, for those of you who don't remember your high school chemistry of what molar, actually it's a totally useless unit. It's roughly molecules per cubic micron. Okay, so 1 nanomolar is 0.6 molecules per cubic micron. So if you want, they can live in a world where the typical concentration of the molecules outside is of order 1 per volume of the cell. And there's a gradient such that if they went 30 microns in this direction, the concentration would go up by 10%, and they can detect that. And they'll bias their motion in that direction. So in this case, there's actually at least 2 kinds of chemical signaling going on. One is that they're simply measuring something about the world outside of them. If you want, the world is sending them a signal, although that's a little too poetic. And so they're just measuring this chemical signal in the outside world in much the same way that we see things or taste them. But then there's an internal chemical signaling in which the way in which events at the receptors eventually modulate the swimming is by changing the concentration of some molecule inside the cell. And so this actually illustrates an important point that cells quite generally, this is a very explicit example, cells quite generally are responding to chemical signals from the outside, but they're also generating internal chemical signals, and they're also very sensitive to those. So in fact, if the concentration of these signaling molecules inside changes by 10%, then essentially the motors all flip from clockwise to counterclockwise. Okay? So a very small change in concentration is enough to produce a very large change in the behavioral output of the cell. Okay? Now what about the situation for genes? So every cell in your body has exactly the same DNA, if all goes well, or rather, more precisely, if nothing has gone wrong. What makes the different cells different is that they choose to read out different parts of the information. So if I look at a piece of DNA, this part of the DNA actually might be the part that actually codes for a protein. So there's some particular protein that you want to make. Let's say suppose that you were a bacterium or another kind of single-celled organism, or one of the cells in your body. In order to do your business, you need a source of nutrients. There are different sources of nutrients that are possible. And any nutrient that you're going to use, you need enzymes that chew up that nutrient and turn it into other useful stuff. But for example, if there's 2 different kinds of sugar, if one of those kinds of sugar is present and the other one not, there's no point in making the enzymes for the sugar that's not there. So you turn those off. You don't actually, the enzymes are proteins. You don't read out the information. You leave it just sitting there, whereas the ones that you actually need, you read out. So how does this work? Well, it's complicated, but a crucial part of the process is that in order to read out the information, it's done in 2 steps. There's a big complex of molecules that bind to the DNA and literally walk along it. And I do mean literally. One can now do experiments on single molecules, observe this walking. And in fact, you can observe it taking steps. As you know, the DNA is made out of a sequence of bases, and you can actually see this molecule walking in steps whose distance is the space between the bases along the spiral of the DNA double helix. And as it walks, it pulls molecules from the surrounding solution and spits out behind it a molecule called messenger RNA. And then this goes to these remarkable factories in the cell called the ribosome, and out of that comes the protein. So if I want to regulate, if I'm a cell and I want to regulate how much protein I make, I've got several chances, right? I could make lots of protein and destroy it, and that way I could control how much I have, but that seems kind of wasteful. I could control this process by which I've got a bunch of messenger RNA around, and I read some of it or not, and that'll vary how much protein I make. But sort of the best thing you could do is to control the thing at the start. So if you don't want to make the protein, don't even make the messenger RNA. And if you do want to make the protein, make more of the messenger RNA. And the way that's done is that close to the place where you have the information to make the protein, you also have special binding sites where other proteins that are floating around in solution can come and bind. And in the simplest picture, you could imagine that there are proteins that bind and stick. They recognize this particular little sequence of DNA, bind here, and prevent this molecule, which is called the RNA polymerase, from actually binding. And in bacteria, that simple geometrical picture actually isn't so bad. In a big, complicated animal like us, not so good. Okay? These regions are bigger and the interactions are more complicated, but the intuition's not bad. So one way you could stop proteins from being expressed is basically have a signaling molecule that recognizes these sequences along the DNA, binds there, and prevents this guy from starting his work. Alternatively, it might be that there really isn't any good place for this molecule to stick, and the only reason it manages to find its starting point is because it touches this blue molecule, which helps it find its way. And so then in that case, the binding of these blue molecules would be a positive effector or an activator for the reading out of the information. Again, this is all happening inside the cell, and in fact, in animals like us, it's happening inside the nucleus, at least this part. This part happens outside. And so what you can think is that the regulation of the reading out of the information, or the expression of these proteins, is in a sense a response to the changing concentration of these blue signaling molecules. Okay? And all of these molecules have names, and I'm going to try and see if I can avoid using them, okay? Because I don't actually think it's essential for what I'm going to tell you. And there's already a little bit of jargon on the board. But that's the picture. And so I hope you realize that there's something similar here, right? That here, as every cell in your body is trying to decide how much protein to make, or how much of this particular protein to make, in a sense, that decision is a reading out or a response to this signal, which is a changing concentration of these blue regulatory molecules. Inside the bacterium, the decision about whether to run your motors clockwise or counterclockwise is a response to the changing concentration of these signaling molecules that I've drawn in yellow, which are themselves a consequence of the response to these molecules in the outside, right? So there's a natural question here, which is that if you're doing all these things inside a cell by changing the concentration of different molecules, how much can you change the concentration? Or let me say it another way, by how little can you change the concentration and have that have a reliable effect? Is it the case, for instance, that the only thing you can do here is have either the blue molecules are at high concentration, all of these sites are filled, and as a result, you're reading out the protein as fast as you can, or you get rid of all the blue molecules, this guy can't get started, you don't make any protein, and it's just an on-off switch? Or is it the case that by modulating the concentration of these signaling molecules, I can fine-tune the level of protein that I make and actually distinguish many different gradations in the signal? Similarly here, can I give a kind of quantitative response to what's happening in my environment? Or am I just sort of stuck in this world of things are good, things are bad, and I have a very coarse response? So the question that's behind all this then is, what's the smallest difference in concentration that you can measure? Okay, now let me be clear, there are other situations in which we know exactly the analogous problem is completely solved, right? So if I try to send a signal to you using light, then I know that the best thing you can do is just count the photons as they arrive. And with a normal light source, the photons arrive as a Poisson process, and so there's shot noise. If you count, on average, n photons, there'll always be square root of n fluctuations. Okay? And that will mean that you can't distinguish arbitrarily small changes in the intensity of the light. So presumably, since here you're counting molecules, there's an analogous limit. You might guess that if you counted n molecules, there'd also be square root of n fluctuations, but you have to think about, you know, what's the right n here, okay? So there's an intuitive argument for this, which was given in 1977 by Berg and Purcell. Berg is the guy who discovered that bacteria swim by rotating their flagella. Purcell is the guy who discovered NMR, or one of the several people who discovered NMR.
How far this kind of analogy just be convincing for the people? You used something related to photons, right?
Right. So
in my view, I don't know why related.
So the reason I brought up the example of photons is that I would say my usual experience is in a physics audience, more people know that there's shot noise in counting photons, that they just haven't thought about the fact that there must be shot noise in counting molecules too. And so it's a sort of nod to an intuition that I hope we share. But now we have to do a real calculation here. So yeah, so simply saying, if you said to an audience of biologists, you know it's like counting photons, that doesn't get you anywhere. Yeah, no, it's not. No, and in fact, if you think about it, the way in which the square root of n effects appear in the case of photons is very different than the way in which, so the way they come to be true in the case of molecules. So it's, in particular, it is possible to produce beams of light which don't have square root of n fluctuations, whereas it's rather harder to produce situations in which counting molecules will allow you to escape from this. So let me give you the intuitive argument due to Berg and Purcell, and it was the following. What they said was, suppose that you build a receptor that has some size A, and effectively it's then sensitive to a volume of A cubed. And the molecules that you're trying to count are present at some concentration C. And so if you count and you just count once, then on average, you're going to get C times A cubed molecules. But we know that the molecules are randomly moving in and out of this volume. They're diffusing around. And so if you take a snapshot and you ask how many molecules are in this volume, then that number will fluctuate by square root of n. So you'll also observe that there are fluctuations, and I'm going to put a 1 here because it's one snapshot, which are of relative size 1 over square root of n, right? So the mean is this. The standard deviation is also this. So if I take the fractional fluctuations, they're order 1 over square root of n. Now, the point is that if you ask about your ability to make a measurement, right, you'll notice that the count is on average proportional to the concentration. So what that means is that in some sense you can interpret the count as being the concentration except for units. So that also means that your fractional error in estimating the concentration will be the same thing. So this will also be delta C over C, again from one snapshot. So how can I do better than this? Well, when you go in the lab and you try to make a measurement, if there's a lot of noise, then what you can do is to average over time to reduce the noise. So how does that work? Well, it works because over time you make many independent measurements of what's hopefully constant across all of those measurements. And you can average them together. The noise will be suppressed and the signal will stay the same. How much do you reduce the noise? Well, you could reduce the noise by a factor 1 over square root of k, where this is the number of independent measurements. Well, how many independent measurements can you make? Well, it's the time that you're willing to average over divided by some characteristic time or correlation time that determines how long it takes for all the fluctuations to jiggle around enough that you really get an independent measurement. Well, in this case, it's pretty clear what that time is. It's the time required for the molecules that are inside this volume to diffuse out and new molecules to diffuse in. Right? If you don't wait for that, then you're just counting the same molecules over and over again. And if you made a mistake the first time, then counting them again might help. But it certainly isn't going to tell you any more about what the concentration is in the outside world. In order to find that out, you need to re-equilibrate. So the characteristic time is the time for diffusion in and out of this volume, which is a squared divided by d, where d is the diffusion constant. And if you put all these factors together, what you get is this, which says that your fractional error is 1 over the square root of a number, and that number is composed out of all the parameters in the problem, in fact, combined in the only way that makes sense by dimensional analysis. Right? There should be a concentration here, but concentration is molecules per unit volume. So in order to get a number, I have to multiply by a volume. How big is that volume? Well, I guess it's bigger if I'm bigger, but that's only 1 power, and I need 2 more powers. Well, the diffusion constant is a length squared per time, and that's how long you're willing to average, and that gives you the right number of powers. Right? Now, this is a fabulous result because it says that for any process in which you're measuring a signal that results from changing concentration, there's a limit to how precise you can be that depends on these very general physical properties of the whole measurement process. Okay? There are no details in here about what happens when the molecule binds to the receptor, or how the binding of these blue molecules to these red sites actually influences the binding of this large complex that eventually reads out the messenger RNA. All details are gone. So this is either incredibly good news for physicists, because it says that you can make claims about the limits to precision of signaling in a biological system without knowing all these details, or it's a complete disaster because we've left out all the details, and we all know that the details in biology are important. Okay. So what my colleagues and I did, which is not, I mean, it's an interesting exercise, but the answer is that the answer is the good answer, right? What we did was we tried to understand, can you do something? This is a very sort of hand-wavy calculation, right? So can I do an honest calculation that gives this answer? So how do you do that? Well, I have these binding sites, and I have this molecule, and it can bind and it can unbind. And then when it unbinds, it diffuses around and then it can bind back. And then when it binds, that can influence the state of some other molecule, and so on, right? So start making a model like that. And then remember that if the system is just sitting here, everything fluctuates, and molecules come on and off, and states of other molecules fluctuate. But those fluctuations are thermal fluctuations, right? Molecules binding and unbinding from their site is a fluctuation in thermal equilibrium. So that means that the fluctuations in this setting should be describable as thermal fluctuations in the same way that we describe Johnson noise in a resistor or Brownian motion of a mechanical system and so on. And so by doing that, you can study a series of increasingly more realistic models and show that if you decide, for instance, that you're going to read out the concentration by monitoring the state of some other molecule, then the precision with which you can read out the concentration of the signaling molecules can be written as essentially a fractional variance, which looks like the term derived by Berg and Purcell, and then there's something which has all the details in it, but importantly is always positive. So what we were able to prove was that for a very wide range of models, if you put the details back in, all that happens is that the noise level goes up, never goes down. And furthermore, since all the parameters are sitting in here, you can imagine that the parameters could be adjusted to make this smaller and smaller until finally this is the dominant term. So what this tells you is that Berg and Purcell really identified a limit to the precision of chemical signaling. And now we can ask, well, how close do real systems come to this limit? Well, the problem that Berg and Purcell started with was actually this problem of bacterial behavior, since that was what Berg was studying. And what they showed was that, in fact, if you use this argument, I mean, you know the concentrations of things because you were doing the experiment, you controlled them. At best, the size of the receptor would be the size of the entire bacterium, which seems sort of generous, but that's the best you could do. You can measure the diffusion constant of things, and then we can discuss how long the bacterium actually averages. And the first thing that they showed was that if you imagine that the way the bacterium decides that this is a good direction in which to swim is by measuring the concentration in its front half and its back half and taking a difference, then it'll never work, because the fluctuations that come from this random diffusion will always be bigger than that signal under the conditions where bacteria successfully find their way. So this shows you that bacteria are basically too small to make a measurement of a spatial gradient. The alternative is to make a measurement in the time domain as they're swimming. They can compare the concentration near the beginning of their run and the end of their run. And since they're swimming at 20 body lengths per second, in 1 second, they cover a distance which is 20 times longer than the distance across their body. And if they're in a gradient, they get a signal that's 20 times bigger. And that's enough to actually detect, okay? But only barely. And the only reason they can do it is because they average for all of that time. And in fact, you can say more than this. You can notice that since they're moving, there's actually a problem. This calculation assumed that everybody was standing still. If you're moving, remember we said that you have to integrate for long enough that you actually get a new sample of the molecules. But remember, if you're moving at a constant velocity, you cover a distance which is proportional to time. But the molecules are diffusing, and so they cover a distance which is roughly the square root of time. For short times, this is always bigger. So that means that if you're trying to make the measurement along your trajectory, you have to wait a little longer in order to be sure that you get a completely independent sample. So there's a minimum amount of time that you need to average for to be sure that you're really seeing something independent. And that time is about a second. And there's a maximum amount of time you should integrate because the bacteria are so small that they actually undergo rotational Brownian motion. And so in about 10 seconds, they're completely disoriented. So you have to average over a time which is boxed in by the physics of the problem to be somewhere between 1 and 10 seconds. And furthermore, you have to be sure that you're actually measuring a derivative along your trajectory so that you don't confuse life being good for life getting better. Okay? Which in the case of bacteria would be disastrous. We can discuss what the situation is for us. Might be that sometimes not worrying about whether life's getting better and appreciating that life is good would be a good thing, but never mind. For bacteria, if they're constantly looking for more sugar, they had better be able to tell the difference between there is sugar around and I'm finding more by going in this direction. And the only way they can do that is that they actually measure the derivative of concentration along their trajectory, not the average concentration. And then they must average that signal for somewhere between 1 and 10 seconds. So Steve Block, who was a student of Howard Berg's, went and measured this by basically taking the bacterium, chop off the flagella, stick the bacterium down by one of the flagella. Now, of course, the flagella are supposed to rotate and push the bacterium, but now I've stuck the thing down, and so what happens is the bacterium rotates, which is also a kind of cool thing to see, but never mind. We've all gotten used to it. And now I can tell whether the motor is going clockwise or counterclockwise to see the whole bacterium moving around, right? And so now what I do is I give a little puff of the chemical that is the sugar that they're responding to, and I just measure the probability, do this many times and measure the probability that it's rotating clockwise or counterclockwise. And you see that this probability is modulated in time after the little puff, and it's a derivative. So the probability, if you give a positive puff, the probability goes up, and then it actually falls down and goes below its average and comes back, right? So if you give a little impulse like this, then you see the probability do that. And this area actually perfectly balance. So it really is taking a derivative. And furthermore, this time scale really is right there in the middle between 1 and 10 seconds. And so the bacterium is both making a measurement which is as accurate as possible given the physics of its environment, and the calculation it's doing, if you will, is exactly the calculation it needs to do in order to make sure that it gets as reliable an estimate as possible. Okay? You can also do, as part of these experiments where you probe how the individual cells are responding while you change the concentration, you can actually try and back out from those measurements an absolute sensitivity. That is to say, we now know how many receptors there are on the surface and so on, and you can show that the gain of the system is such that you really are getting a significant modulation in the probability of going clockwise or counterclockwise when one more receptor, on average, is occupied by a molecule. And so it really is true that essentially the system is counting every single molecule and making every molecule count. It's actually modulating its behavior. And one way of thinking about the Berg per cell limit is to realize that this combination, D times A times C, is the rate at which molecules are arriving into this volume as a result of diffusion. So if you imagine that this volume were a perfect sink so that any molecule that arrives here disappears, the rate at which molecules would be disappearing is D times A times C. Okay? So that means that that's the rate at which molecules are entering the volume. And so this is like the thing with photons arriving, right? The rate at which molecules are arriving is this combination. This is the number that you count, and therefore this is the square root of n fluctuations that you see, because the molecules are also arriving there. Okay? So so this subject, in a sense, started with thinking about the case of chemotaxis. But once you understand that the limits are quite general, you might ask about other examples. And so my colleagues and I have spent a fair bit of time trying to think about this example of gene regulation in the context of embryonic development. So let me tell you a little bit about how. Yes, yeah.
Is there a way to interpret the action potential in the same way? Because the picture reminds.
No. This has nothing to do with it. That's it's not. No. This one has a simple no. Right. So this was the probability of going counterclockwise. This was time, and this was the concentration of the molecule outside. So let me give you sort of a different example of this chemical signaling that comes from this case of gene regulation. So let me talk about a fruit fly, which is one of the biologists' favorite examples of embryonic development. Embryonic development's a little different in every organism, and there are some big differences among different classes of organisms. Let me just talk about flies. So the egg of a fruit fly is about 0.5 millimeter long. And you probably know that the end result of embryonic development of a fruit fly is not a fly. It's a maggot, right? In the same way, somewhat more charmingly, the result of embryonic development in a butterfly is a caterpillar, right? Caterpillars are more charming than maggots, but it's the same idea, okay? Right? There's another stage of metamorphosis later, which we're not going to talk about. But many of you will have looked at these things, right? And you will know that maggots and caterpillars and so on have segmented bodies, right? So if you just look at a caterpillar, you can see that the body comes in segments. Okay? A really extraordinary discovery was that if I look at the embryo of the fly 3 hours after the egg has been laid, and I measure the concentration of particular proteins, what I discover is that some of these proteins have concentrations that come in stripes. Let's try something else. And so what this means is that the cells have actually decided, I'm a cell sitting here, and so I'm going to make the pink molecule and not the yellow one. And I'm a cell sitting there, and I'm going to make the yellow molecule and not the pink one, and so on along here. Now, if you came away from the talk and you thought, wow, that's the blueprint for making the segments, that wouldn't be exactly right. It's a little more complicated than that, but it's not far off. Okay? And in fact, the very first thing that happens, and I'll actually show a movie of this at the talk for a general audience tomorrow evening, is that you make sort of a furrow in the sort of ball of cells right about here to separate the head from the rest of the body. Okay? And that really does happen at the boundary between these two stripes. Okay? Plus or minus one cell. Okay? It's very precise. So the first observation, which didn't have to be true and is kind of marvelous, is that there really is a blueprint. And you can read this blueprint within hours after the egg has been laid.
How does that 3 hours compare to embryonic development?
24. So it's, the maggot walks out after 24 hours.
Is there time for a cell to divide?
At the beginning, when you start with 1, get to 2, and so on, the cell duplication time is running 10 minutes or less. It's running, as far as anybody knows, flat out, right? You're duplicating as fast as you can. In fact, it's going so fast that in the fly or in insects, unlike most other organisms, you don't even have time to make membranes that divide the cells. So what you're doing is duplicating your DNA and making more nuclei, but you haven't only at about 3 hours, everything stops and you build all the walls between the cells, and then you read out this blueprint and start moving everything around. Okay, so what does this have to do with the problem that we have here? Well, what it has to do with it is that how does the system know where to put these stripes? So one possibility is that the system starts out being completely symmetric, and there's actually an instability to produce this spatially periodic pattern, as Turing proposed in 1952, right? Exactly 50 years ago, and half of the centenary to his life, right? Sorry, 60 years ago, because he, of course, sadly didn't live to be 50. Sorry, 60 years ago. However, for better or worse, that's not what happens. Because it turns out that the mother has broken the symmetry before any of this happens. The mother, when she makes the egg, actually places the messenger RNA for particular proteins at cardinal points in the egg. And what happens is that once the egg is laid, well, all of them, but let me focus on this one. Let me do one because it's easier to think about. This messenger RNA begins to be translated into protein. And so what you have is a big box, and you have a source of protein at one end. So what's going to happen as you make that protein, it's going to diffuse out into the rest of the box, and perhaps it'll be degraded along the way. And so after a while, if I monitor the concentration of, so let me call this molecule number 1, if I monitor the concentration of protein number 1 as a function of position, I'll see something that looks like this. And you can observe this either by stopping the action and making antibodies against this protein, tagging the antibodies with a fluorescent molecule, and mapping out this concentration profile. Or, since this is the modern era, you can genetically engineer the fly so that whenever it makes this molecule, it makes it attached to a fluorescent molecule, and then it'll just glow live, okay? Which is pretty impressive. In fact, my colleague, Thomas Greger, who's been doing these experiments, has now gotten to the point where he can even do that fluorescent protein trick with some of these striped things. So you can actually see this striped pattern rising in real time, which is quite extraordinary. So the reason that all of this is connected to the problem that we've just been talking about is that if I have a cell sitting here and a cell sitting here, then if this gradient in the concentration of this protein number 1 is really the primary signal that tells me where I am along the length, I have the problem that these two cells see concentrations which differ by only about 10%. So one possibility is it's all much more complicated than this. But the other possibility is that there's a real problem here, right? Because we also know that these molecules are, in fact, these blue signaling molecules that bind to the DNA. And what they do is to turn other genes on and off, and that's ultimately how you get this striped pattern. But if you think about it, for gene regulation, the size of the target is a few nanometers. It's this little piece of DNA that you're aiming for. So let's say it's about 3 nanometers. The diffusion constant of proteins in the cytoplasm has been measured. You can either measure it for this particular protein or for others. And you get numbers which are on the order of microns squared per second. Then you can ask, well, what's the concentration of these signaling molecules? Well, again, you want the concentration to be somewhere in the range where, if the concentration is a little higher, then mostly these sites are filled. If it's a little lower, mostly they're empty. So the real question is, at what concentration do these binding sites tend to be about half filled? And so that has to do with the physics of the way the protein interacts with the DNA, and this is something people have measured. And so we know that these concentrations are, again, measured in nanomolar, which is to say molecules per cubic micron. So if you put all of these together, DAC is about 3 times 10 to the minus 3 per second. And so that tells you that delta C over C would be something like 300 seconds divided by the averaging time. Well, 300 seconds doesn't sound so bad. That's 5 minutes. But in 5 minutes, you would only, according to the Berg per cell limit, be able to distinguish concentration differences which are order 1. So if the concentration doubled, you'd notice. But smaller than that, you wouldn't. To detect a 10% difference, you would need to integrate for 100 times this long. And now we're talking about many, many hours. Okay? And you've only got 3 hours for the whole thing, and surely you don't use all of that. It's probably 10 minutes near the end that really matters. So what's the solution to this? Well, one possibility is that really the system isn't this precise. So what my colleagues did was to start doing experiments where you measure not just the concentration of this protein, but also the concentration of one of these proteins that's being directly regulated by it. So you should imagine you take the cell, you take the embryo, you stop the action, which means you slightly cook the egg, actually. And now you add antibodies against 2 proteins. One is the signaling protein that's supposed to regulate the expression of the gene, and the other is the protein that's made when the gene gets turned on. And now in one image, you can look and see thousands of cells, and you can plot for every single cell if I measure the concentration of protein number 1, which is an input, and then I measure the concentration of protein number 2, which is the output, I can see that on average kind of looks like this, which sort of makes sense. This is what you get if several of these molecules are binding to a group of sites here that all interact cooperatively. But then you can also measure the noise level. And then you can turn the noise level into an equivalent noise level in the input. And sure enough, this is about 10%. By the way, in fairness to my experimentalist friends, it's extraordinarily difficult to do this measurement. Remember that if it's really true that cells are responding meaningfully to 10% changes in concentration, then you better measure the concentration much more accurately than that. And it's not so long ago that people assumed that you never needed to measure the concentration of a protein to better than a factor of 2 accuracy. So the fact that you now have to, that this shows you that it's worthwhile and in some sense necessary to make measurements on the scale of a few percent is sort of shifting all this away from that sense of sloppiness that we started with. So we're still left with a paradox because this precision really does seem to violate this rule. But remember that the fly is special because there are actually no walls between the cells. And so in fact, molecules that are made by one nucleus can diffuse to another nucleus. And if you know the diffusion constant of these molecules, you realize that in 5 minutes, you can diffuse through an area that has about 50 cells in it. And so in a sense, the response of one cell or one nucleus reflects an average over about 50. And, well, 60 converts hours into minutes, right? So you can trade averaging over more cells for averaging over more time. And you can then just barely make it. Okay? So then you say, well, nice story. How do you know it's true? Well, if the cells are communicating with each other and averaging, then their fluctuations become correlated. Whereas if they decide independently, of course, the fluctuations are not correlated. So in the same image where you saw all these fluctuations, you now go back to the image, from every cell, subtract the mean. You now have a field of noise, compute the correlation function in the field of noise, and you see that there's a correlation length which is just large enough to make up for the averaging time. So this provides you with evidence that, as in the case of bacterial chemotaxis, this chemical sensing in bacteria, the fly's embryo is making measurements which are very close to the limits that come from the random arrival of molecules at their targets. And in fact, it's only the special features of the fly embryo that make it possible for this to happen in this case. Okay? It really leans on everything matching up just right in order to get this much precision out of the system. So what I'd like to do, and what I'd like to close with, so normally if I think about this, for me, I've been trying to understand these kinds of problems both in the context of these chemical signaling problems and in the context of signal processing in the nervous system. And so for me, the nervous system problems came first. And so usually when I talk about this, I start there. Here I decided it might be interesting to talk first about the molecular case. And a consequence of that is that the time for the neural case has sort of disappeared. Let me make just a quick observation. I mentioned at the beginning, well, I've mentioned a couple of times now, that this sort of shot noise in the arrival of molecules, kind of like shot noise in the arrival of photons, in the same way that these systems are, in effect, counting every single molecule. Your visual system is capable of operating in a regime where it's counting every single photon that arrives at your retina. And there's a whole variety of questions that people, my colleagues and I included, have looked at in trying to understand how it's possible to measure these single photon signals reliably. And there's a whole host of problems, one of which is that when you measure, so in these cases, right, if I change the concentration of molecules by 10%, well, that, you know, it might be only 1 or 2 molecules, but it's also 10% of the molecules. In the cells in your retina, there are 1 billion molecules of rhodopsin, which absorb the light. And when one of them absorbs a photon and changes its structure, that's enough for you to see something, or for the cell to respond and contribute to your perceptions. So the problem is very different. You have to be sort of sensitive in this absolute sense to single molecular events. And single molecular events are random, right? So not only is the arrival of the photon random, but anything that this molecule will do is random. And so in particular, if it goes around and triggers some biochemistry, then you have the problem that it lives for a length of time, which is itself a random number, and thus the amount of catalysis that it does that produces the internal signal in the cell will be random. And so if you're not careful, the response to single photons will vary enormously. And so part of the way that the cell solves this problem is that the process by which rhodopsin turns itself off, right? It gets turned on by absorbing the photon, and then it's alive for a while and can go around and do a variety of things. Part of the way it turns itself off is through these phosphorylation reactions that I was telling you about at the beginning. And if there was one phosphorylation step, then there would be an enormous amount of shot noise associated with the time at which that step occurred. The fluctuations would be very large. Instead, it turns out that there are, at least in many species, 6 phosphorylation sites. And essentially, you by putting several random processes in a row and waiting until you get to the end to say that that's when the process is done, you turn these random molecular events into something much more like a clock. And so what looks like unnecessary complexity is actually there to regulate the noise, you know, to push the noise level down so that you can actually count single photons reproducibly. Okay. So the last thing I want to say, I mean, this was sort of an aside, right? Just to realize there's a whole other body of examples. The last thing I want to say is, over here we were able to say that if you want to reach this level of precision, then the bacterium has to do a very particular computation, and you can then check to see whether it really does that. So what we'd like to do is to do the same thing over here and say, well, if the embryo wants to make measurements, as it were, of these primary spatial gradients as accurately as possible, what is it that it has to do in order to actually reach this level of precision? And so that's a question you could answer at a lot of different levels. One thing that we've done is to ask, suppose I take these noise levels as given. Okay? Is there anything else the system can do to optimize its performance? Okay? It's already apparently squished the noise level down as low as it can get. What can I do that's left? Well, the answer is sort of obvious. If the inputs were always down here, then this wouldn't be a very useful device because the system would always be off. Similarly, if they were always up here, that wouldn't be very useful. If they were confined to a very narrow dynamic range here, that would also not be useful. What you'd really like is to have the range of inputs make full use of the range of outputs. And then you would transmit through this regulatory unit as much information as you possibly could. So what you'd like to ask is not, I mean, there's one problem, which is what's the noise level. But then if I tell you that I know the noise levels, so essentially I've been able to characterize the probability distribution of the output given the input. So this is about noise. I still can ask how much information gets transmitted between the input and the output. And that depends both on the input-output relation and the noise, but it also depends on the distribution of inputs. So I could ask, how do I maximize this by adjusting the distribution of inputs? And the answer is that if the noise level were homogeneous here, then what you would like to do is to use all of your available outputs equally often. Because then so you can think about it that there's a noise level. The noise level effectively produces a kind of quantization that says there's only certain levels you can distinguish. The best you could do then is to use all of those levels equally often. You'd maximize the entropy of this discretized output. In fact, that can't be true because these are molecules. Down here, if you actually are making 0 molecules, you also have no variance. Up here, you're making lots of molecules, and you're not very sensitive to your inputs, so your relative variance will actually be small. In here, the variance is actually quite large because you have this steep input-output relationship, and you have this noise in the arrival of the input molecules. So the noise in this system is actually quite structured. What that means is that if you follow through this theory and predict, for example, the probability distribution of the output, which is a little more interesting because the distribution of output is sort of more intimately tied to the structure of the noise. If you plot this like this, then you discover that there's a non-trivial prediction, which is something like this, which tells you that you might think the system is basically an off-on switch, but the intermediate levels are predicted to be used with some non-zero probability, which is related to the details of the noise in the system. And so now you go back to those images where you had stopped the action and could look at every cell, and you measure this probability distribution. And you discover that it agrees perfectly with the theory. Okay? So what this tells you is that not only has the system pushed the noise level down as far as it can, it then that it's made, if you want, as good a communication channel as it can for taking information from signals to expression levels of genes. It now sculpts the distribution of signals to get as much information through that channel as possible. The problem with this analysis, which makes us think that you can't just declare victory having gotten this right, is that this is pretending that there's one input and one output. And really, there's a whole network of interactions. So we could argue that there are reasons why focusing on this one transformation is useful. But we could also ask, how do we generalize this to think about the whole network? And the way to do that is to say, well, really, what's happening here is that there's position where the cell is on the embryo. This gets turned into the concentration of this primary signaling molecule by the mother. The mother does this for you, basically. And then this gets turned into, and maybe there's even a C1 prime here and a C1 double prime, right, that we're not keeping track of. And this gets turned into a collection of proteins which together form a network. And we actually know what these all are. They're they're sitting in a sort of hierarchy just above those fancy stripe patterns. They make somewhat coarser patterns, and they're called gap genes because if you're missing one of them, there's a gap in the pattern. And so what you can ask is, if I had to try to optimize information flow from here all the way to here, is there an analogous prediction? And remember, the prediction is that you should use, if the symbols had equal noise, if there were equal noise levels, you should use them equally often. So if the different combinations of concentrations of all these proteins produced equal errors in estimating position, then I should use them equally often. But I could turn this around, right? Because I know that the different positions are used equally often. The cells are spaced equally along length. So in some sense, if I go to an arbitrary point and I measure these concentrations and I try to infer my position, the accuracy should be constant, right? If I have this matching condition obeyed. And so that experiment's now been done by one of the graduate students in Thomas Gregor's group at Princeton, Julien Dubuis. And it works. And it works in a very subtle way because the spatial patterns of all these genes are very complicated. But somehow, and the noise levels are complicated, but somehow they conspire with each other so that the accuracy with which you can estimate position is independent of where you are along the axis. And that's exactly what you would expect if you were optimizing information transmission all the way from here to here. Furthermore, that accuracy is actually 1%, which is the accuracy with which things are positioned along the axis, from the long axis of the embryo. So what have I tried to tell you? I've tried to tell you that although there are lots of things about biology that are incredibly variable and complicated, if you dig into particular examples, you can sometimes see extraordinary precision, whether that's in the modulation of the swimming behavior of bacteria or the modulation of the expression levels of genes in the embryo. Here you're seeing responses, reliable responses to 10% changes in concentration, and the modulations of the output are again accurate to 10% or better if you're up here. These noise levels are down very close to the limits that are set by counting the individual molecules as they arrive randomly at their targets. And so you can't do better than that without using more molecules. And presumably, you'd need more molecules are expensive, so that's why you don't do it. And then once you've pushed your noise level down, you can still do more, which is to try and push as much information through the system as possible. And that leads to, at least in the first round, predictions about the way in which you use the different symbols in your code, if you want to think about it that way. And those predictions seem to be in accord with experiment. So what we would really like to do, of course, is to do more than this and say, let me take the principle that I'm trying to maximize my information and not just say something generally about the nature of this transformation and these distributions, but actually talk about the real network that underlies all of this and say that I can predict something about the form and structure and even detailed quantitative things about that network. And that's one of the things that we're trying to do, but it's very much a work in progress. And as I mentioned, there's a sort of parallel story in the context of signal processing in the nervous system where you're talking not about measuring changes in the concentration of molecules, but measuring changes in light intensity or measuring motion across the visual field or something like that. And many of these same ideas have echoes. And in fact, this idea about matching the distribution of inputs to the input-output relation in noise is an idea which, in fact, was first used in the context of coding in the nervous system, where people tried to sort of understand the way in which the input-output relations of neurons are matched to their distribution of inputs. But of course, the distribution of inputs in the world varies, and that predicts that the input-output relationships of neurons should vary, and that's a well-known phenomenon. It's called adaptation, and this path is a path to a quantitative theory of those adaptation effects. So I hope to have convinced you that as I say, there is precision to be found. That exploring that precision is at least gets us on the start of a path to something that starts to look like a theory in the sense that we are used to that word in physics. So thanks very much.
Questions?
Bill, suppose you had a gradient of glucose, let's say, in the x-axis, and suppose you had a gradient of other kind of food on the y-axis. How does the E. coli decide how to weight the two vectors to give you an average direction? And also, is the noise independent if you do that?
So it's interesting. If you go back to Berg's original work from the early to mid-1970s, he has a few remarks about what happens if you give the bacterium mixed signals. And then there's nothing. And so I really don't know why people haven't gone back to this. I think it's a really interesting question. There's sort of two parts to the question. One is the kind of mechanistic version of the question. Well, you know, everybody says there's a signal here and a signal there, and they're supposed to add together. I mean, you've only got one set of motors, right? So you can only do one thing. So, I mean, you might have internal signals that allow you to keep these separate, but in terms of behavioral output, you can only do one thing. So one question is, do the usual cartoons actually work? But then there's a more subtle question, which is, you know, what should the bacterium do? And is it sort of smart enough to know? You might wonder, for instance, whether, you know, part of the history of chemotaxis, as you probably know, is the discovery that this really is separate from metabolism, right? You might think that the reason you know to swim toward more sugar is you sort of just check internally how healthy you are, and if there isn't enough food, you should try going somewhere else. But that's not true, that you actually sense the sugar concentration. But of course, your metabolism is controlled in part by which genes you express. So you might think that you should also, I mean, there's no point in swimming toward a sugar that you can't yet metabolize, right? So you might think those were coupled, and there's no reason to think not, but nobody's checked.
That's just one obvious question about what you said, and then a couple of questions about the fly. I mean, there's a messenger inside your little blue circles, and do different receptors for different sugars and nutrients tend to produce the same messenger?
So, so in bacterial chemotaxis, there's several different kinds of receptor. But for this, there's only one internal signaling molecule that talks to the motor. So they all go through that bottleneck. The question is whether they do something non-trivial on the way, right? Do they just add up their signals, or do they do something a little more sophisticated?
A number of different types of receptors.
Right, but for example, the receptors are all fairly densely packed, and they certainly interact with each other.
So now coming back to here, so one question is, one sees these strides, and one might look for some periodicity, or one might say, well, you know, there's really, you know, protein 1 that does the first stride, protein 2 that does the second stride.
So, I think the most, so maybe the way to phrase this is, you know, I have this intuition from Turing that says I know how to make periodic patterns. So do I emphasize that this is periodic, or?
So you got already quite a bit of the mechanism, and in fact, periodicity would seem to be a hard thing to detect at this point.
So so you know what's happening is, you know, here's molecule number 1 and here's molecule number 2, but there's also a 3 and a 4 and a 5, and actually there's even this, and then these guys inhibit each other, and all sorts of terrible things happen. So now, and then all of this feeds into the thing that makes the stripes. So what you can do is, let's go to the piece of DNA. So it's interesting, you know, the development of experimental techniques determines at least the order in which we find things out, okay? You might be even more worried that it seems to determine what people think actually is happening. Okay, right? Let's not, okay, it's not philosophy of science. Let's just talk about what the sequence in which things happened. Our ability to manipulate DNA exploded around the time that this was discovered. So there's amazing experiments by my colleague Eric Rieshaus and Johannes Volhard where they identified all of these genes. Okay, and as Eric likes to say, if he said, he believes they did a really good experiment and that was a smart thing to do. Said, but the reason they really became famous is that while they were doing this, other people were developing recombinant DNA, right? Because otherwise they would have had a bunch of bottles on the shelf with mutant flies, each of which was mutant in one of the genes that's relevant, and they'd sit there they could have sat there for 50 years, right? Nobody would know what to do with it. In fact, at the same time, other people were figuring out, well, if I have a mutant, I can pull the thing out and I can sequence the gene and I can do all this other stuff. So let's take the gene that codes for one of those stripes, one of the proteins in those stripes. What you discover is that this region, which I made a little sketch of, really huge. And you can reach into that region and say, well, let me knock out this little piece. And then stripe number 2 disappears. And then let me knock out some other piece, and stripe 4 and 7 disappear. And so on. And so what's going on, presumably, is that this gene is being turned on by some very complicated combinatorial rules of different proteins. I mean, presumably, it's these proteins that are binding, right? But in some very complicated logical way. And so you should think of this, the patterns of on and off as a sort of complicated Boolean output, as opposed to focusing on the periodicity.
There's not some sort of a priori periodicity, but it's not happening with a different set of proteins. It's happening within the regulation.
Now, to be fair, it's also true, of course, that the cells can talk to each other. And in that process, they could establish some of the scales.
You could have gone back and postulated that before, and you found it didn't seem to be needed, except in this very diffuse way. So my other kind of more simple question is that it seemed like, if I kind of grant your figures, very generally in this kind of developmental mechanism, the time scale has to be 30,000 seconds. Things can't happen faster than like a day. It's just these special cases like the insects where somehow one gets around. So that's a fair statement.
Well, I wish I knew that for sure. So for instance, you can also do experiments which are in some sense the union of bacterial migration and developmental biology. You can study the development of neurons, which of course, in order to wire up your brain, they have to find their way to the right place. So it's developmental biology like this, but it's about cells migrating like this. And there too, they're extraordinarily sensitive to small gradients. I mean, they are following gradients, and we know what the molecules are. And you can do controlled experiments and show that they are in fact pushing down near these same Berg per cell limits. Except, of course, it's all very slow. And in fact, there's something which I find visually very compelling, but I certainly don't know enough I don't know enough numbers to plug in and see if this is right. There are sort of major decision points as neurons migrate. So for example, the cells that are coming from your eyes, I mean, you know that, right, the right side of the world is represented on the left side of your head, and the left side of the world is represented on the right side of your head. But each eye sees both halves of the world. So what that means is that half of the cells in the optic nerve in your right eye have to go this way, and half of them go that way. And so they're growing up, right, and they reach some point where they have to do this split. So, I've got a bunch of axons coming out here, a bunch of axons coming out here, and they come to this point. So some of these go this way, and some of these go that way, right? If you watch them growing, right, the leading edge of the cell always has this kind of ruffly thing that looks like it's exploring. When they reach this point, they just stop. So they were extending at a relatively constant rate, and when they reach this point, they just stop and start exploring. And they pause there for a really long time, and then they go. And I find it very hard to believe that this is not because they need a really reliable signal to decide which way to go, and the only way to do that is to integrate longer. But I don't know an experiment that tells me that's actually correct.
Well, let me throw out another speculation. on that, which is that one asks, what is the difference between the human and the chimpanzee set? And of course, one focuses on things like the brain is big. But the other comment you could make is that, well, the human takes 10 years, to develop its brain. The chimpanzee. Yeah. So you have that time to actually wire the thing properly instead of.
I don't know that. Yeah, I don't think that's quite fair. I mean, there must be. I mean, there's an analog of this everywhere, right? So if I'm trying to set. I mean, let me look at it another way. Suppose the brain has wired, but of course all of the synaptic connections have numbers attached to them, the synaptic weights or strengths, and all of those have to be adjusted. So the question is, how many of them can you manipulate, and how precisely can you do them? So you have to. So you presumably need to gather enough information over a long period of time, right? If I want to adjust more of them to the same accuracy, and not just have them be copies of each other, I better collect more information, because what am I using? What numbers in the environment am I measuring that tell me how to set all these parameters? And so the longer developmental time allows you to. So if all you were doing. Sorry, let's be clear about this. If all you were doing was making a bigger brain, in the sense that bigger animals have bigger brains than smaller animals, because, for example, if I want to process all the data coming in from my skin, if I have 1 million nerve fibers coming in from my skin instead of 100,000, I guess I need more neurons. But that is more of the same. And so it doesn't actually take me longer to measure all the things I need to measure in order to figure out how to wire it up. But if I really want to use the full power of gathering more data, or if I want to take the same data and discover higher order structures in it, then I need to integrate for longer in order to measure those things. And so, not unreasonable. Jin.
So Bill, is your maximization of the mutual information a proposal, or you are trying to think as a principle?
So we're trying it out as a principle, right? I mean, so what can I tell you? I can tell you that it sounds like a sensible thing to try, and the things that we've tried, we've tried to derive predictions from it that seem to be working, and we have tried. Obviously the goal is, can I derive this with all the numbers on the arrows from this principle? And we don't know how to do that yet, but we do know how to do pieces of this problem. Do I know for sure that this is the right principle? Well, no, of course not. But so that was the spirit, right? Was to convince you that by starting with these ideas about noise, I could eventually find my way to something that starts to feel like it might be a theory that has enough in it that I could keep calculating and keep checking, as opposed to checking one number here and checking one number there. And so I think we're there in the sense that we started with this, and then we went to this business of measuring multiple genes, and now we're trying to see if we can derive things about the structure of the network. It could collapse next month, right? But that's good, right? It's good that it can be wrong. Yes.
So a lot of signal involves cascades, which is a special type of network which seems to go against this idea that the information is really behind it. Because in a cascade, at each step, you lose information. So how come that instead of just getting the information from the input directly to the output, you routinely go through 3, 4, 5 steps?
So in photoreceptors, this is also true, right? The rhodopsin molecule changes the structure in response to the photon. This couples to a member of the G protein family, which then couples to the phosphodiesterase, which reduces the concentration of cyclic GMP, which eventually binds, or rather doesn't bind, to the ion channels in the cell membrane. In that case, you need this in order to generate gain. And so it's from the noise part of the problem, right? That I can't get enough gain to overcome the other noise sources in one stage. Now it's interesting actually to compare chemotaxis and photoreception. In both cases, in some sense, you're sensitive to single molecular events at the beginning. But at least in our current understanding, to which I have not contributed, so I'm reporting, in chemotaxis, this sort of enzymatic gain is there but not dominant. The dominance seems to be in the sort of cooperative effects of the different molecules. Whereas in the case of the photoreceptors, it seems like it's the cascaded enzymatic gain which is critical. And I mean, that's, if you take the literature at face value, that's what you get. And I must admit that I don't have an understanding of why some systems should be one way and other systems the other, but that seems like a really good question.
So you talked about photoreceptors in the retina as an example in the nervous system. So do you think this can apply also to higher brain functions, like decision making?
Yes, it's a long discussion, but yes. And so one of the things that we're trying to do actually, so the problem here, and I would say this is true, there's a more general observation to be made, which is that in all of these cases, right, it's pretty obvious what's supposed to be signal and what's supposed to be noise. Yeah. And it's also obvious that if you're going to capture more information about what is also pretty obvious, right? You really want to know where you are, because that's what determines what you're supposed to do, right? I think the problem in moving from sort of simple brain functions to more complex brain functions is that one loses this sense of information about what. So one of the ideas that we're trying out, and again, this is the beginning of a long discussion, but just to give you a flavor, we're trying out the idea that the really important thing is about making predictions. So there's information, I could gather information about lots of things, but the one thing I know for sure is that there's no point in gathering information that doesn't have predictive power, because I can never use it to guide my actions. So maybe what I always want to do is to extract the information that's relevant about the future of whatever my inputs are. And maybe I just iterate that, right? So you imagine that everywhere in the brain, whatever your inputs are, you're always, the computation you're always trying to do is to extract the information that's predictive. And that you can do anywhere, right? And as you do it deeper into the brain, where you have longer feedback loops, you end up extracting, presumably, information that is predictive on longer and longer timescales. Okay? And in fact, you can show that even some aspects of learning can be cast into this framework of extracting predictive information. So we think that there is an information theoretic principle that can be carried from the sensory periphery all the way to higher level things. But at the moment, that's sort of, it's an idea, right? And we actually started by going back to the sensory periphery and asking ourselves, are neurons in the retina actually extracting features which are the ones that have predictive power? Which is not the question people usually ask. And the answer seems to be yes, that it really is, if you ask which bits about the past are these neurons extracting, the answer is they're extracting the bits that have maximum predictive power. So we think that this is a path, but we're at the beginning of the path. Okay.
Thank you very much.
Thanks.